Beginning in the 5th week, primordial germ cells migrate from the umbilical vesicle to the gonadal ridges, bulges which are formed from proliferation of the epithelial and underlying mesenchyme, medial to the mesonephros. The germ cells travel along the dorsal mesentery to reach their destination. During the 6th week, these cells are incorporated into the gonadal ridges. At week 7, the developing gonads of males and females are identical in appearance and are referred to as being indifferent. In males, the gonads develop into testes. This requires a Y chromosome complement. The SRY gene codes for testes determining factor. Under the influence of TDF, the original undifferentiated gonadal ridges differentiate into seminiferous cords, which will become the seminiferous tubules. The cords branch and connect to form the rete testis. A fibrous capsule, called the tunica albuginea, also develops and is characteristic of testes formation. The rete testes become connected to the efferent ductules, which in turn connect to the duct of the epididymis, derived from the mesonephric tubules. The wall structure of the seminiferous tubules contains two cell types: Sertoli cells, which support the development of sperm, and the primordial germ cells or spermatogonia that will begin to mature into sperm during puberty. The seminiferous tubules are separated from each other by mesenchyme that produces interstitial or Leydig cells. By about week 8, the Leydig cells begin to produce testosterone and androstenedione. These androgenic hormones induce masculine differentiation of the internal sex organs and the external genitalia. For instance, the mesonephric duct becomes the ductus deferens. In females, the two X chromosomes provide genes that help to regulate ovarian development. The development of gonadal cords and rete ovari are rudimentary and normally degenerate over time. Cortical cords extend into the developing ovary from the surface and the germ cells incorporate there. At 16 weeks, these cords begin to disintegrate into cell clusters or primordial follicles, each containing an oogonium or primary oocyte. The follicles are surrounded by follicular cells derived from the surface epithelium. Active mitosis of oocytes occurs in the fetus, producing additional follicles. No primary oocytes develop postnatally. Without the influence of the Y chromosome, the mesospheric ducts disintegrate, while the paramesonephric duct contributes to a number of structures in the female, including the uterine tubes and the uterovaginal primordium. Initially, the development of the reproductive ducts is similar in males and females, with the formation of bilateral mesonephric ducts and bilateral paramesonephric ducts. The undifferentiated gonad can also be distinguished. The mesonephric ducts exit into the urogenital sinus, while the inferior ends of the paramesonephric ducts fuse to form the uterovaginal primordium, also connected to the urogenital sinus. In males, the testes secrete both androgenic or masculinizing hormones as well as Mullerian-inhibiting substance, MIS. The Mullerian-inhibiting substance causes the regression of the paramesonephric duct, while androgens promote the formation of male genital ducts. The mesonephric duct forms the epididymis and the ductus deferens. In this close-up view of the developing male's secondary sex glands, we see the paramesonephric duct beginning to regress. The prostate begins to form from outgrowths of the urethral endoderm, while seminal glands form as outgrowths of the caudal ends of the mesonephric ducts. Lastly, bulbourethral glands develop from the spongy urethra. In the absence of a Y chromosome and under the influence of the X chromosome, the female reproductive ducts begin to form. The fused inferior portions of the paramesonephric ducts, the uterovaginal primordium, can be seen entering the urogenital sinus. Over time, the mesonephric duct involutes and the paramesonephric duct differentiates into the uterine tubes, the uterus, and the superior portion of the vagina. The fusion of the paramesonephric ducts also creates a peritoneal fold that becomes the broad ligament and also creates two peritoneal compartments, the rectouterine pouch and the vesicouterine pouch. In the early stages of development, the form of the external genitalia of males and females appears similar or indifferent. We see the genital tubercle, the large labioscrotal swellings, and the smaller urogenital folds. The cloacal membrane has not yet ruptured at this stage. By week 7, these structures continue to grow and the anal membrane is now differentiated from the cloacal membrane. In males, and under the influence of testosterone produced by the testicles, the genital tubercle forms the glands of the penis, while the urogenital folds fuse along the ventral surface of the penis to form the spongy urethra. Similarly, the labioscrotal swellings grow and fuse in the midline, forming the scrotum. This fusion is marked by the scrotal raphe. As the urethral folds fuse, the urethral groove is formed and the spongy urethra sinks below the surface. The overlying surface ectoderm fuses along the midline, marked by the penile raphe. The corpus cavernosum and the corpus spongiosum develop from mesenchyme of the early phallus. At the tip of the penis, an ectodermal ingrowth forms a cellular cord which extends toward the root of the penis. This cellular cord becomes canalized and joins with the spongy urethra. This brings the urethral opening to the tip of the penis. A ring of ectoderm also forms at the peripheral aspects of the glands. This ectoderm undergoes partial apoptosis, resulting in the formation of the prepuce or foreskin. In females, the development of the external genitalia begins with the same undifferentiated structures. However, without the influence of testosterone, the genital tubercle becomes the clitoris. The labioscrotal folds become the labia majora, with a small fused superior portion becoming the mons pubis. The urogenital folds become the labia minora. By the 9th week, the external genitalia of the fetus begins to show distinguishing characteristics, and by the 12th week, the external genitalia is fully differentiated. In males, the inguinal canal provides a path for the testes to descend into the scrotum. This process is associated with the growth of the embryo, the atrophy of the paramesonephric ducts, and the influence of testosterone. The process vaginalis is an evagination of the peritoneum that herniates through the abdominal wall, carrying with it the layers of the abdominal wall which form the coverings of the testes and spermatic cord. In females, the ovaries remain in the abdominal cavity, with the blood supply contained within the suspensory ligament, a remnant of the mesonephric duct. The round ligament of the uterus passes through the inguinal canal and attaches to the labia majora.